Good morning, everyone. Welcome back from spring break. We'll be doing something a little bit different since we're doing these lectures remotely. We're going to be chunking these up a little bit. So I've included places where we can stop, and those will also be indicated on your handout as well. So I know it's been a long time. You guys have probably slept a few times in the spring break interim there. So let's begin today's lecture with a review of what we did before spring break. All right, so most importantly, glycolysis. We spent a lot of time learning about glycolysis. So just as a reminder, this is how your body converts glucose that you eat into energy in the form of ATP and NADH, and other useful intermediates for other pathways, the most important of which is pyruvate. So there at the bottom of the slide, we've got the equation we're all familiar with, glucose plus 2 molecules of NAD plus and 2 molecules of ADP and 2 inorganic phosphorus, or inorganic phosphates, excuse me, react to form 2 molecules of pyruvate, 2 molecules of ATP, 2 NADH, and 2 hydroniums. So, don't forget this takes 10 steps. It's not a one-step reaction. And this is what you get on net. So remember, you have to add 2 ATP molecules in and you get 4 ATP out. But on net, you get 2 ATP. So glycolysis can be really split up into 3 main phases. The energy investment phase, the phase where you break that 6-carbon sugar into 2 3-carbon sugars, and finally energy generation, where you get that energy out more than what you invested in the first third of glycolysis. And pyruvate, as the end point of glycolysis, can be used for other pathways as well. Of course, we know about the Krebs cycle, which is the most important one, but there are also other alternate pathways, such as fermentation to form ethanol or lactate formation. And the main reason each of these exist is to regenerate NAD+. And if we don't have any NAD+, we can't continue with glycolysis. So the main point of these is to regenerate that important cofactor for that enzymatic pathway. So the most important pathway for pyruvate is the Krebs cycle. This is where a lot of NADH will be formed. So pyruvate is first converted to acetyl-CoA before entering the Krebs cycle. And carbon from acetyl-CoA is converted to CO2. Though if you'll remember, from acetyl-CoA, it's not converted directly into CO2 on the first go-around. It has to participate in the cycle once before those particular carbons get converted to CO2. And this pathway is regulated via feedback loops. So if you have too many high energy molecules, too much NADH, for example, the Krebs cycle will slow down, or it will work to slow down glycolysis, thereby slowing down the Krebs cycle. This way you don't have any reactions that go out of control. So the theoretical maximum for the complete metabolism of glucose is 38 ATP. If you've learned in other classes that it actually generates less ATP, that's probably the more scientifically accurate thing. Reason being, some of the energy consumed in the metabolism of glucose gets used for other things. That's not for ATP generation or NADH generation. But theoretically, at maximum, you could get a yield of 38 ATP from one glucose. Kind of a lot. So today we're going to be talking about oxidative phosphorylation, which is tied in with the Krebs cycle and glycolysis. So we have 6 total learning objectives for these lectures. 1, we're going to define oxidative phosphorylation, or OXPHOS if you want to save some time. Next, we're going to describe where each step of OXPHOS occurs within the mitochondria, or within the cell. We're going to categorize high energy molecules and transporter molecules, describe how a proton gradient is formed in oxidative phosphorylation, and how that proton gradient is used to generate ATP. And finally, we're going to make sure to identify uncoupling in the context of OXPHOS and why that can be dangerous. Okay, let's get back to starting about energy balance. So this is where your handout is going to start at. So we talked about how ATP is a power outlet for the cell. You've got to have a constant concentration of ATP in order for your cell to be happy and do the things that it does on a regular basis. And we talked about how the Krebs cycle makes a lot of NADH and other high energy compounds. And we talked about how 1 NADH equals 3 ATP molecules. But why is that? How does 1 molecule of NADH become 3 ATP? Why can't we say that it's worth 3 ATP? If you'll think back to what we were talking about in class, how ATP is like making change from a $100 bill. You can't just spend NADH at a store. You've got to break it down first. So oxidative phosphorylation is how we convert NADH into ATP, how we break down that $100 bill of NADH into to make change, basically. So the pathway that oxidizes electrons from the Krebs cycle is called oxidative phosphorylation. NADH and FADH2 are the mobile carriers from the Krebs cycle that get oxidized. Remember, these are hydride carriers, so they have an H plus and 2 electrons. And those electrons are what get oxidized to make that energy, to make that ATP. Another thing you might remember is that when I was teaching the Krebs cycle, I made sure to focus on QH2. FADH2 gets turned into QH2, and today we'll talk about how. So oxidative phosphorylation is actually responsible for most of the ATP generation in cells. We talked about how the Krebs cycle makes a lot of NADH, and those are equivalent to 3 ATPs. But this is the pathway which actually makes that ATP from NADH. So this is where most of the ATP generation in cells occurs. And the really interesting thing, in my opinion, is that the intermediate in this pathway is not a specific chemical. You don't have a, ADP plus inorganic phosphate forms ATP. You don't have actual chemical intermediates. You have this physical proton gradient. So just because you have more protons outside of your membrane than you have inside your membrane, that leads to a chemical potential energy. And the cell uses that potential energy gradient to actually make something awesome. So this is a little bit complex, but really kind of fascinating when you think about it. We've been through a lot of pathways that have these definite chemical intermediates, and now we have a pathway where we have a physical thing which is acting as an energy intermediate. Anyway, I'll stop philosophizing about that. The mechanism by which this proton gradient gets used as energy intermediate is called chemiosmosis. So chemicals, in this case ions, protons. Osmosis, you probably already know this, movement across a membrane. So the movement of those ions across the mitochondrial membrane is what allows it to be used in ATP generation. Okay, so let's clarify FADH2 really quick. So NADH, nicotinamide adenine dinucleotide. Cool, we know this one. FADH2, flavin adenine dinucleotide. So FADH2 gets converted into QH2, so it's kind of a lot like QH2 for our purposes. It's a heavy-duty NADH, and it's got multiple redox states. So it can catalyze particularly difficult redox reactions. But for our purposes, what we really care about is that it turns into QH2. Fun fact, if you look in your book, the reason they're called flavin adenine dinucleotides are because they're yellow. And it says that the reason they're called flavins is because like a house in Rome, like ancient Rome, where everyone had like blonde hair. So flavin means blonde, which is like yellow. So they're called flavins. Works for me. Okay, let's go ahead and get started. So oxidative phosphorylation, or OXPHOS, refers to two separate processes, oxidation and phosphorylation. Pretty intuitive based on the name. In isolated mitochondria, these processes are coupled, which means they happen hand in hand. You can't have phosphorylation without oxidation. And if you have oxidation, you will get phosphorylation. They're coupled. For example, adding ATP or ADP, excuse me, causes oxidation of the substrates. So these are coupled in a mitochondria. They're two separate processes, but if you have one, you'll get the other. Or if you have phosphorylation, you have to have oxidation. All right, so the overall electron flow in this is from NADH, from say the Krebs cycle, to Q, which is, as you remember, ubiquinone, to O2, to oxygen. So NADH gets oxidized to NAD+, and O2 is reduced to water. So this is sort of a fine point. So we all know we breathe in oxygen and we breathe out CO2. And the intuitive thing to think is, okay, so I breathed in oxygen and I stuck a carbon onto it and I breathed it out. But as we've learned, that's not exactly the case. The CO2 comes from processes like the Krebs cycle, but the oxygen actually gets consumed in oxidative phosphorylation. So the oxidation or the consumption of oxygen is not directly tied to breathing out CO2. They happen in different processes. But anyway, overall, oxidative phosphorylation consumes oxygen and produces water. So during OXPHOS, the movement of electrons and protons are coupled, and we'll talk more about that. So the movement of protons and electrons occurs within the inner membrane of mitochondria. So as you know, mitochondria have a membrane, and the electron movement takes place within the membrane. Throughout the process by which electrons go from one electron carrier to the next one until finally they turn oxygen into water, electrons will move through the membrane, but protons will get moved across the membrane. They'll move from the matrix, which is to say inside the mitochondria, outside the mitochondria to the cytosol. So matrix is the space within the mitochondria, and cytosol is the space within the cell, but outside of the mitochondria. So you can see this makes an electrical and chemical gradient across the mitochondrial membrane. If you keep pushing positively charged H+ outside of the membrane, you'll make a negatively charged space within the membrane. So, you know, take plus from in the matrix and keep dumping it into the cytosol. Eventually, you'll have a whole bunch of positive charge outside the membrane, and what's left in your membrane will be negatively charged. So yeah, so this is literally like a battery, actually. So the main takeaway, what you need to know about oxidative phosphorylation: oxidizing NADH exports protons across the membrane, forming a charge gradient. If you keep pushing protons across the membrane, you will make a difference in charge across that membrane. That's what gradient means here, a difference in charge. And re-importing these protons synthesizes ATP. You push a bunch of protons out, and if you want to synthesize ATP, you just got to let them back in again. You open the door and let them flow back in. So here's a picture of what it generally looks like. By the way, we're now on page 2 of your handout. I didn't include this picture within your handout because at this point it's not very informative. But you can see it's going to be kind of a confusing process. There's 5 different enzyme complexes that you can identify, plus this CYT thing over here outside of the membrane, and a bunch of arrows inside of the membrane. So I hope by the end of this lecture series, you'll be able to identify what's going on in each of these. I know it's confusing now, but let's work through this and try to clear some things up. First, let's look at the mitochondria. You know, as I'm sure you've all heard, the mitochondria is the powerhouse of the cell. So that's a very, you know, sort of blasé thing to say. But why is it the powerhouse? Why is it the powerhouse of the cell? Why does oxidative phosphorylation happen here and not somewhere else? So let's take a look, actually just look at a mitochondria. So here it is on the right. The outer membrane can be seen as a dark outline in this micrograph. But if you look at the inner membrane, it's got many foldings, many like a zigzag type of structure. And this provides more surface area. Well, more surface area, as we know from chemistry, is more space for reactions to occur, more space for those membrane-bound enzyme complexes to occupy and do their job. So it's got a big zigzag structure, a really high surface area. So in the mitochondria, we have the outer membrane, which encloses it, and there are large pores. You might remember this from the Krebs cycle. There are porins, which allow things to diffuse into the mitochondria. And then there's an intermembrane space in between the outer membrane and the inner membrane. And remember, as we said, the inner membrane is what counts for OXPHOS. If you look in your book, they actually say that you can strip away the outer membrane of a mitochondria and it still works just fine. The inner membrane is what really matters. So the inner membrane has a really high concentration of proteins, not just enzymes, but also carrier proteins, things to actively transport different molecules from outside of the mitochondria into the matrix, into the inside of the mitochondria. And these protein complexes control the flow of protons and electrons to set up that charge gradient. So let's start off small. Let's start with the electron and proton carriers. Later on, we'll move into the enzymes and proteins that really do the heavy hitting in oxidative phosphorylation. So this will be starting at the top of page 3 on your handout. So first, let's look at carriers of electrons. So of course, metals such as iron are electron carriers. You know, the wires in your house are made out of metal, and those carry electrons. Those transfer electricity from the power plant to your house. Well, they don't work any different just because they're in a cell instead of in your house. If you have an iron molecule or a copper molecule, for example, or sorry, a copper atom, those can carry electrons just as good, every bit as good, if we're making a malto meal joke here. I hope somebody gets that malto meal joke. So for example here, we've got a heme group. Heme's not just for blood these days, or any days. But that iron at the inside of heme can carry electrons. And these are called chelated. They're grabbed onto by a protein group. Or they can complex with sulfur. You can have iron which reacts with sulfur to make these little clusters, and those can also grab onto electrons and transport them. So in general, these will mediate single electron transfers. We also have carriers of protons. So proton, as you might remember, H+. So these are going to be literal acids and bases. If you can move a proton, you're a carrier of a proton. The most common ones for here are going to be aspartyl and histidyl residues of proteins. So these can carry protons. And we'll see one of these later on. If you want to go back to chapter 3 and check on the structure of these, it should illuminate why these are proton carriers. Finally, we have carriers of both electrons and protons. So these are the ones you know and love: NADH, QH2, FADH2, and FMNH2, which is another flavin related to FADH2. These carry a proton in the form of H and electrons in the form of, well, electrons attached to that hydrogen right there. These are typically called hydride transfers, I suppose. So that's an H-, which would be an H with 2 electrons on it. I really don't like the term hydride transfer because hydrides are generally pretty reactive. Though for what it's worth, these are pretty reactive too. In any case, you can see they have an H, and they also have some high energy electrons attached with them. So these are carriers of both electrons and protons. Oh yeah, I guess I already talked about this. So NADH transfers electrons along with protons, and FMNH is a flavin cofactor, just like FADH2 is. And these can transfer electrons either 1 or 2 at a time. So dang, we've got a whole lot of high energy molecules. We've got, of course, ATP, ADP, AMP. That's the high energy molecule we're trying to make here today. all the ones we've talked about today. Of course, we've got GTP, which is a different nucleoside. So let's go ahead and try categorizing these. All right, I hope you've taken a few minutes to categorize things. Let's go ahead and go over these, get that information down. All right, here's what I came up with. So sorry about the handwriting, though it's probably better than what I do on the whiteboard or on the chalkboard on a daily basis. So for proton carriers, we've got aspartyl and histidyl residues, as we just talked about. For electron carriers, we've got iron-sulfur complexes and metal ions. And both things that can transfer electrons and protons are going to be NADH, FADH2, FMNH2, and QH2. And the tricky thing to remember is that ATP, ADP, AMP, and GTP are neither proton nor electron carriers. They are high-energy phosphate transfer molecules. So they're sort of cousins to the rest of these, but they're not technically, they don't mediate proton transfer, for example. So that's what's maybe a little bit more difficult to remember. That's what I want you to take away from this activity, how to categorize each of these molecules. So, because we're all learning this at home due to the current circumstances, I thought it'd be nice to sort of chunk this lecture up into smaller parts. So I think this is a good stopping point. This is about a third of the way through the lecture. So, go ahead, get your notes arranged, take a breather, and then come back and we'll start into membrane-bound complexes and electron pathways. All right, welcome back for part 2. So for oxidative phosphorylation, there are 5 membrane-bound protein complexes that participate in this, and easily they're named complexes 1 through 5. Biochemists are really creative when it comes to naming things. So here's a picture of complexes 1 through 4. So this entire situation we've got here is a pathway of connected redox reactions. So those electrons are getting transferred from NADH to Q to oxygen. And this is in order of increasing reduction potential. So the opposite of that would be steadily oxidizing flow. So this is a way you can view that. NADH is the pathway substrate. This is what we start with in order to get the pathway started. And water is the pathway product. At the end of the day, oxygen gets converted to water. So here's a picture. You don't need to memorize anything about this, but I think it should get the point across where the electrons are going in oxidative phosphorylation. So NADH to Q, to this thing that's called cytochrome C, which we'll talk about, all the way down to oxygen. And each of these steps is mediated by one of the enzyme complexes. So complexes 1 through 4 are oxidation, and complex 5 is ATP production, phosphorylation. So this particular pathway is supported by scientific evidence, which is great because we're in a science class. So in order to figure out where this pathway goes, where it starts, where it stops, and what's in between, you can use inhibitors. Previously, people have asked me, well, how do biochemists know that the pathway looks like this? If you can dose a cell with a specific inhibitor that prevents one complex from stopping, the other complexes downstream of that come after that complex will also be stopped. So you can sort of do a process of elimination type of thing. So for example, if you put rotenone into a cell, that'll inhibit complex 1. So no oxidative phosphorylation will occur. Instead, if you hit a cell with malonate, that'll inhibit complex 2. So complex 1 will work just fine, but those electrons will stop at complex 2. So you can say, okay, so complex 1 comes before complex 2. Same thing for antimycin or cyanide. So by adding inhibitors, that can support the sequence as we know it. Or, sorry, that doesn't support the sequence, that provides support. These inhibitors are not involved in everyday oxphos. So electron carriers closer to oxygen are more oxidized. That should be pretty easy to remember. The closer you get to oxygen, more oxidized. Carriers on the NADH side are more reduced. So more oxidized, closer to the end of the line, more reduced, closer to the beginning. Okay, so let's take a deep breath and jump into talking about each of these complexes. So this is the bottom of page 4, the start of page 5 on your handout. So complex 1, the first proton pump. This is, well, a proton pump is an enzyme complex that takes protons from within the matrix of the mitochondria and pumps them into the cell out of the mitochondria. So proton pump. And its formal name, I guess, on its birth certificate is NADH reductase. And it accepts electrons and protons in the form of a hydride ion from NADH. So as I was talking about hydride carriers, you can see that it transfers NH, hydrogen, as well as 2 high energy electrons. So those high energy electrons bounce around within this enzyme complex that go to FMN, to an iron sulfur complex, and then finally to the mobile carrier Q, ubiquinone. So remember, those electrons from NADH get transferred to ubiquinone. And you might notice that Q right there is inside the membrane. So Q is a pretty hydrophobic molecule, so it likes to be inside membranes. So cells take advantage of that. And if you want to get some electrons inside the membrane, they get transferred to Q. So as it transfers these high energy electrons from NADH to Q, becoming QH2, it pumps protons across the membrane during the process. So these things are linked. The electrons move into the membrane, the protons move across the membrane. So this enzyme complex has dozens of subunits. It's huge. And it contains bound flavin, as you can see with the FMN right there, and lots of little iron sulfur complex mini proteins. So we don't need to know the huge structure of the first proton pump of complex 1. Just know that this enzyme complex is indeed pretty complex. It's got a lot of parts to it. So we will dive into this just a little bit. You don't need to memorize the individual proteins within. There are just too many. But you should know that for complex 1, there's the N module, which is the input domain. So this is where NADH comes in and passes those electrons through different electron carriers. So N module, NADH module. This is the input part. P module is a transport of proteins across the membrane. So, P is for proton. And the way it does this is via a conformational change. I don't know if you remember how GLUT1 works back in glycolysis, but you might remember that when a glycolysis binds, it undergoes a conformational change and flips around. So the glucose is now within the cell. So a lot of different transporters work like this. Once something binds, it undergoes a conformational change and flips around. So this is how transport of protons happens across the membrane. It's not the only way that it can happen, but for complex 1, that's how it happens. Finally, we have the Q module, where the electrons react with Q, ubiquinone, to form QH2. So 3 major parts. We've got the input part, the N module, the proton transfer part, the P module, and the Q part, the Q module. So that was complex 1. You can see it at the far left of this picture right here. NADH gets input and those electrons get transferred to Q. Meanwhile, a proton is pumped across that membrane. Next, let's look at complex 2. So this is going to be on starting in, I guess, the bottom of page 5, the top of page 6 on your handout. So complex 2 is succinate dehydrogenase. And you might remember this. This might trigger your memories for the Krebs cycle before spring break. So this is the only step in the Krebs cycle which is membrane bound. Well, this is why, because involved in multiple pathways. It's also a part of OXPHOS. So it's sort of a tool for multiple reactions within the cell. But the sequence of reactions is similar to complex 1, but this time we're not starting with NADH, we're starting with FADH2. So when I talked about the Krebs cycle about how QH2 is formed, that's because FADH2 from the Krebs cycle basically goes right in to complex 2 here and becomes QH2. So electrons get passed from FADH2 to these iron sulfur complexes within complex 2 and passed to Q. No protons are translocated in complex 2. This is not a proton pump. Complex 1 pushed protons across the membrane. Complex 2 does not. All it does, it takes electrons from FADH2 and puts them onto Q. So you can see between complex 1 and complex 2, we make a lot of QH2. Both of these take different electron transporters and put them onto Q. So let's go ahead and use up that Q. Let's make that QH2 good for something. And we're going to do that with complex 3, shown in the middle here. So complex 3 is the second proton pump. Its formal name is Q-cytochrome C reductase. Good name. And what happens is it takes those high energy electrons from QH2 and it reduces cytochrome C. So what is cytochrome C? It's a small heme-containing protein associated with mitochondria. You can see a picture of it here. There are various cytochromes. We'll actually run into a little bit of them later on. But cytochrome C is directly involved in OXPHOS. So this small protein is actually essential for getting OXPHOS to go. It's the substrate for the second proton pump. So an interesting thing about this is that the electron flow is partially cyclic. It's called the Q cycle. So it undergoes single electron transfers only for this pathway. So the reduction of Q all the way down to fully reduced QH2 takes 2 steps. You don't transfer 2 electrons at once. You transfer 1 electron and then another electron. And the reason this takes place in 2 steps is because it increases the reaction efficiency. It's a little quirk of the cell. It makes the transfer more efficient. Okay, so you can see that in complex 3, we do pump a proton across the membrane. This is also a proton pump. But the substrate for complex 3, cytochrome C, the thing that gets reduced, is actually sitting on the surface of the membrane right there. So it's outside of the membrane. That'll come important later. Okay, so finally complex 4, proton pump and annihilation. So this is the final complex in electron transport. So oxidation, that's 4 protein complexes. It takes a lot of work. Phosphorylation just takes 1 protein complex. Enzyme complex. So this is the final complex in electron transport, and it has 13 subunits in mammalian cells. So it is, once again, it's a real huge complex. And it's also known as cytochrome C oxidase. So that cytochrome C that we just reduced, we're going to oxidize it. And here the pumping of protons is also tied to the conformational change. So here's a picture. You can find this also at the top of page 7 in your handout. So cytochrome C, which is sitting there on top of the membrane, transfers electrons down to a copper atom, which then get transferred to something called cytochrome A, cytochrome A3, and finally down to oxygen. So you can see there within the matrix, oxygen plus 2 protons become 1 molecule of water, so half a molecule of oxygen, I guess. Meanwhile, while this occurs, 1 proton gets moved from within the matrix to the outside into the cytosol. oh yeah, I guess I talked through most of this. Overall, 4 electrons flow through the complex, making 2 water molecules. So from one cytochrome C, you can get 2 water molecules. And remember, cytochrome C is not consumed. It's oxidized and reduced. So it's not getting destroyed. It's just getting changed. Its oxidation state is just getting changed. So it is, in reality, a very efficient way to transfer electrons. So there are 2 mechanisms for generating this proton gradient so far. Pump mechanisms, which are related to that conformational change that we've been talking about. If you literally pick up a proton inside the mitochondria and put it outside, that's going to generate a proton gradient. That's going to generate a difference in the concentration of protons. But there's also one more way that you can generate that proton gradient. If you lose protons on one side of the membrane, that's equivalent to gaining protons on the other. So for example, if you do a redox reaction with cytochrome C, which is outside of the membrane, and the protons in that came from inside of the membrane, that's also equivalent to a pump. It's not literally throwing protons outside of the membrane, but what it is doing is basically doing an accounting difference. If you lose protons on the inside, that's chemically equivalent to gaining protons on the other. All we care about is the difference in concentration. Okay, let's summarize. Complexes 1 through 4. The overall result is that a proton gradient is produced across the inner mitochondrial membrane. So complexes 1, 3, and 4 are proton pumps. They send H+ outside of the membrane. So these are the ones that set up your proton gradient. Complex 2 is not literally a proton pump itself, but it does generate QH2 from FADH2. So along the process of generating this proton gradient, electrons from high energy molecules such as NADH get transferred all the way down to O2, generating water. So we've got 3 complexes that are proton pumps, and while these pump protons outside of the membrane, we transfer these electrons all the way down to oxygen. So this is another good stopping point. Go ahead, take a pause, get your notes together, write down any questions you might have, and feel free to email me them. And when you feel like you're ready, maybe on another day even, I don't mind, go ahead and step ahead to the final portion of oxidative phosphorylation. All right, welcome to part 3. Today we're going to be, or maybe it's the same day even, but today we're going to be talking about complex 5. So we set up this proton gradient to, move those protons outside of the cell, but we didn't make any ATP. We just converted a lot of oxygen into water. So what do we do with this proton gradient? Well, we've exported all these protons outside of the membrane. What if we let them back into the membrane? So if we do this, we can use this motion of protons to drive the synthesis of ATP. So this is literally like a battery. In a battery, we set up a charge gradient. We move some charges around within the battery. And then if you hook up something to that battery, you can use the motion of the the motion of electrons to make your thing go. So the movement of that charge actually can be harnessed to generate ATP. So just like in a battery, movement of charge can be used to do something important. So complex 5, ATP synthase. This one, the name sort of makes sense. It synthesizes ATP, ATP synthase. And what it harnesses for energy is that movement of protons back into the mitochondria. So this complex involves it includes a little passageway for protons to travel through, and as the protons travel through, they give it the energy to synthesize ATP. So it's got a passageway for protons and an active site for synthesis of ATP. Actually, it has 3 active sites for making ATP. So the overall reaction is shown at the bottom of the slide right here. ADP plus inorganic phosphate plus 4 protons outside of the membrane get converted to ATP plus water plus 4 protons inside of the membrane. The protons weren't actually involved in the reaction. They just got moved outside of the membrane into the membrane. And later on, we'll talk about how this actually works. So here's what complex 5 looks like. It's a little bit strange. I guess all proteins look a little bit strange though. So it has a stalk, this lollipop structure here, which is responsible for ATP formation. So that's what's going to be identified in red right here on this particular image, the stalk. And the rotor, identified in green, is what's responsible for the conformational change. And that conformational change, as the protons flow through the rotor, through the green part, is what's going to drive the ATP formation in the pink part, in the stalk. So let's talk a little bit about the mechanism. Let's get a little bit down into the details. Binding of ATP displays negative cooperativity. So if you generate a molecule of ATP, it's going to inhibit any further ATP molecules from being made on complex 5. So the rotor part of complex 5 includes a protein segment. As it spins, it literally kicks out an ATP in order to reset that enzyme complex. So this is kind of neat. We're including a bunch of physical processes into this chemical reaction to generate ATP. So this particular conformational change where we kick out ATP resets the enzyme, and it's key to making the reaction proceed. So let's take a minute and watch this interesting YouTube video which shows why rotational motion of the rotor drives ATP formation. So after you watch it, you should be able to see how the proton gradient gets employed to turn the rotor, and why turning that rotor is really important for making ATP. All right, I hope you liked that video. I think it was really interesting. You can see as the protons move through the channel, that makes the rotor spin. And as the rotor spins, it kicks off ATPs like a little engine. So it acts sort of like a little water wheel in a sense. So let's talk about mitochondrial membrane transport. The intermembrane proteins are also essential for transporting the small molecules. So one good example is adenine nucleotide translocase. Translocase moves something. And here it's going to be ATP from the mitochondria to the cytosol. We just made this ATP using complex 5, and we're going to move it to the cytosol. This is metabolically irreversible. Oh, by the way, this is the bottom of page 8 on your handout. So phosphate exchange is mediated by 2 transport proteins. These move inorganic phosphate around so they can actually make it to complex 5 and react to form ATP. So the pH cotransporter, this draws on the pH gradient. This also uses that battery power that we set up in order to move phosphate. We also have the inorganic phosphate malate transporter. Once again, you don't have to memorize the exact names of these, but you should know that the 5 complexes in OXPHOS can't work on their own. They require these helper complexes to move stuff around for them. If you want to think of it like a factory or like a system of factories to generate ATP, those factories can't run without the trucks that bring them the raw materials. So you can think of these as delivering the raw materials for the reaction to happen. Oh, of course, there are other transport proteins as well. So these, for example, calcium. Calcium is really important for this kind of thing. Okay, so we're at the end of oxidative phosphorylation. We went through each of these 5 protein complexes. So we have 1 through 4, which take the high energy electrons made in the Krebs cycle, and they transfer them all the way down to oxygen, which becomes water. And in doing so, they move a bunch of protons outside of the mitochondria. Finally, complex 5, which you can see here on the left, generates ATP by allowing those protons to come back into the cell. Just like in a battery, movement of charge is harnessed in order to make something happen. So these are coupled. If you were to add NADH and some inorganic phosphate to mitochondria, oxidation would really click up. More NADH equals more oxphos, which means more oxygen used. So they're coupled. Oxidation and phosphorylation are coupled in a mitochondria. And this is indirect through a proton gradient. Once again, it's not through one individual molecule. It's through this battery that we set up, this separation of charges in the proton gradient. So we call this coupling. What about uncoupling? If we break that link between oxidation and phosphorylation, this is called uncoupling. So this sounds ominous. We'll talk about that a little bit. How can this happen? So for example, ionophores can do this. So ionophores are ion carriers. These compounds catalyze ion transport across hydrophobic membranes. So imagine if you spent all this energy moving protons outside of your membrane, and then some garbage protein picked up those protons and threw them right back in without making any ATP. That would uncouple oxidation and phosphorylation. You would not make any ATP even though you oxidized all these high energy electrons. So yeah, this can happen in real life. This is a real thing to think about. A lot of pharmaceutical drugs are hydrophobic, and this helps them enter the cells for therapeutic purposes. Cells have that hydrophobic outer membrane. But if they uncouple oxidative phosphorylation, this can be dangerous. If you can't generate any ATP, you'll die. So if you mess with oxphos, that can be really dangerous. So every new drug needs to be tested for uncoupling. Yeah, so really the major takeaway is if you can't generate ATP, you'll die. And if you uncouple oxidation and phosphorylation, you can't generate any ATP, so you'll die. So let's summarize this final portion here. Complex 5 uses the proton gradient that we set up with complexes 1 through 4 in order to generate ATP from ADP. Excuse me. And as you saw in the video that I linked, it operates sort of like a water wheel. The movement of those protons powers the rotor to kick out each new individual ATP molecule, allowing new spaces to synthesize more ATP. There are various transport proteins that help in getting these mobile cofactors in and out of the mitochondria. Oxidation, which is the first 4 complexes, and phosphorylation, which is complex 5, are coupled. You can't have one without the other in a normal cell. If you uncouple these, that means you consume energy without generating any ATP. All right, please let me know if you have any questions. It'll really help if you add a slide number next to your questions, so that will help me know exactly where you're having trouble. I will send out some quiz questions to go along with this, and yeah, keep at it. I'm sorry about the certain circumstances, but I'm always here to help answer questions if you need it. Please don't hesitate to ask. Thanks, and like and subscribe, I guess.